Hello, and welcome to our webinar on direct-to-consumer genetic testing. My name is Nina, and I'm here with Pam, and we are Master of Health Science students in the Medical Genomics program at the University of Toronto. Today, we will be teaching you how to interpret your results from direct-to-consumer genetic testing companies like 23andMe and Ancestry. We will also draw your attention to a few caveats in your report results. Let's start with explaining what direct-to-consumer or DTC genetic testing is. DTC companies allow consumers to gain access to their genetic data without having to go through a healthcare provider. The process is very simple. You order a DNA kit online and it's delivered directly to you in the mail. It usually contains a collection tube for a saliva sample. Your saliva contains white blood cells, and these cells contain your entire genome sequence, which is all the DNA that makes you, you. When you're done providing your sample, you mail the kit back to the company, and once it arrives, they can analyze your genome in three main ways: SNP genotyping, next-generation sequencing, and whole genome sequencing. The genome is made up of 4 letters: A, T, G, and C. Each of these letters is called a nucleotide. Our genome is made up of 3 billion pairs of these nucleotides, and humans are the same at about 99.5% of them. The other 0.5% is variable in the general population, meaning people can have different nucleotides from each other at those positions. The variable positions are called single nucleotide polymorphisms, SNPs, or variants. SNP genotyping is the most common DNA analysis method used by DTC companies today. During SNP genotyping, they look at selected SNPs in order to gain more information about the things that make us different, such as your risk for developing breast cancer or whether or not you have a widow's peak. Next-generation sequencing is a way to look at every nucleotide at selected locations in the genome. This allows them to find even more SNPs than SNP genotyping and may lead to more precise results. The last type of DTC genetic test is whole genome sequencing. Instead of just looking at SNPs or selected regions of the genome, these companies look at the entire genome, and this allows them to see all the SNPs. For the purposes of this webinar, we'll be focusing on SNP genotyping. When the analysis is complete by whichever method your company of choice performed, all you have to do is log on online to check your results. There's all sorts of information that you can learn from your report. You can find out whether you're a carrier for a recessive disorder, see your risk for developing a complex disease, get a detailed ancestry report, and have some fun looking at the traits influenced by your genes.
When you get your results back, your job now is to read through them and understand the implications they can have for you. To get a better understanding of the report, you will need to know some basic genetic concepts which may or may not be explained in the report. Here, we will explain some of those concepts and go over a few example reports in order to give you the knowledge you need to interpret your own report. As a note, the reporting style varies by company, so 2 companies can report on the same condition but present the results in a different way. Let's introduce 2 basic genetic concepts first. In your genome, there are 2 copies of each gene. Each copy is called an allele. One allele is inherited from your mother, and the other is inherited from your father. This means that for any position in your genome, you get 1 nucleotide from your mom and 1 from your dad. The combination of the nucleotides from mom and dad is called a genotype. There is a set of diseases called recessive disorders. For a recessive disorder, if you inherited an allele from one of your parents that is known to lead to disease, you're said to be a carrier of this disease allele. But is that enough to get the disease? No. Having 1 out of 2 disease allele is not enough to cause disease. But if you inherit the disease allele from both parents, then you will get the recessive disorder. If you are a carrier, you then have 1 in 2 chance of passing down the disease allele down to your children. If your partner is also a carrier, then there are 4 possible combinations of genotypes that your child can inherit: a healthy allele from both mom and dad, a disease allele from both mom and dad, or a healthy allele from one parent and a disease allele from the other parent. In the latter case, the child will be a carrier, but unaffected. Taking all the possibilities into consideration, the child would have a 1 in 4 chance of developing the disease. For this reason, many people want to learn about their carrier status for recessive disorders in order to be aware of the chances that their child will be affected. Here is a quick question for you. If you are a carrier and your partner is not, your child will still be a carrier. Is this statement true or false? Please take a few seconds to think about it. Now let's review the answer. In this scenario, it actually depends. Let's take a close look at this. Like before, if you're a carrier, then you have a 1 in 2 chance of passing the disease allele down to your children. If your partner is not a carrier for the condition, you can only contribute healthy alleles to your children. In combination, the child will have a 2 in 4 chance of carrying the allele, but 0 chance of getting the disease. And if you don't know whether your partner is a carrier or not, it would be harder to predict for your children. One thing to note is that if you do have the concern for recessive disorder in the family, please consult with genetic counselor for professional advice. Let's take a look at an example. Here is a cystic fibrosis carrier status report from 23andMe. Cystic fibrosis is a recessive disease caused by mutations in the CFTR gene. It results in mucus building up in the lungs, recurrent lung infections, and progressive difficulty breathing. Here on the left, person 1's report shows that she is a carrier for CF, since she has one of the disease variants this test looked for. She has 1 in 2 chance of passing this on to her children, and if her partner is also a carrier, her child would have a 1 in 4 chance of developing the disease. On the right, however, the result shows that person 2 does not have any of the variants tested. However, this does not rule out the possibility of him being a carrier. For this person, there is up to a 1 in 230 chance that he has a disease allele for CF. For this particular disease, the tested variants are most prevalent in certain ethnicity groups, which are Europeans, Hispanic or Latino, and Ashkenazi Jewish people. The DTC genetic test is only able to detect and report on variants present in their databases. Depending on the database used, the company may only be aware of some genetic variants that cause the disease. One of the main reasons for this is the fact that most genetic databases contain genetic information from European individuals, meaning that the database will have a larger collection of known variants for European people. Their database wouldn't have as many of the variants causing the disease in non-Europeans, making this test less accurate for people from non-European populations. Another possible reason is the fact that the databases contain the most common variants. If you have a common variant, there is a high likelihood that it will be detected by the test. If you have a disease allele that is more rare in the population, it may not be present in the database, so the test may not be able to detect that. This means that person 2 may still have a disease allele for CF, although the report said there were no variants detected.
The second type of result you can get from your DTC genetic test is the risk for developing complex diseases, such as specific cancers, diabetes, and Alzheimer's. Complex diseases are a little bit more tricky than recessive ones because they're often influenced not only by one gene, but by many other genes as well as your environment and lifestyle. Some examples of lifestyle and environmental factors are diet, exercise, medications, and exposure to mutagens. DTC companies are able to give less accurate results than they can for carrier status because there's so many more factors influencing your disease risk. Therefore, your test result cannot tell you whether or not you will get a complex disease. It can only estimate your risk of getting it. Because no complex disorder has any one variant that makes it so that you will certainly get the disease if you have it, it's very important to keep in mind that just because you have a specific variant doesn't mean you will get the disease. On the other hand, though, just because you don't have a variant detected by a test, it doesn't mean you won't get the disease. Are you wondering how they know which variants to look for? That would be a great question. DTC companies often draw from existing literature to decide which variants to look for. One of the main ways disease variants are found in the literature is through case control studies. If they know a specific gene or set of genes is implicated in disease, they can set up one group of people with the disease, called cases, and one group of healthy people, called controls. They can then look for differences in disease genes and use them to estimate the risk of disease. Another common method used to determine which variants can affect the risk for a complex disease is GWAS, or Genome-Wide Association Studies. GWAS are kind of like case control studies, just on a larger scale. They use a lot more people and look at the whole genome. This is because the variants found by GWAS often only have a small contribution to disease, and therefore they need a lot more people to be sure of their findings. Just like with the carrier status reports, an issue with case control and GWAS studies is that people of different ancestral backgrounds may have differences in the variants they carry in disease genes. Since individuals of European descent are overrepresented in the databases used to determine which variants are disease-causing, the variants from other ancestral backgrounds may not be as well studied. This means that for non-Europeans, many DTC tests may not predict disease risk as accurately as it might for people of European descent. Let's go through an example together. In this example, the consumer was found to have a variant in the MYH7 or MYBPC3 gene. The result shows that they have an increased risk of developing cardiomyopathy, a heart condition that can lead to irregular heartbeat and heart failure. However, there is no quantification of that increased risk. This can make it hard to understand how much the risk is increased and cause heightened anxiety. In the report, they highlight that this test result doesn't ensure that the consumer will get cardiomyopathy. However, it's best to report this finding to a healthcare professional who may be better able to inform you about the actual risk this variant may confer to you. The complex disease reports can also have implications for your family. If you have a variant that increases your predisposition to a specific disease, it's very likely that one of your parents passed that variant down to you and is also at risk. There is also a 50-50 chance of passing that variant down to your children, putting them at risk as well. This report shows the inheritance pattern that occurs when one parent has a variant putting them at an increased risk. It shows that your child would have a 1 in 2 chance of having the condition. However, Pam and I agree that a better description of a case like this would be that there would be a 1 in 2 chance of your child having a higher risk of getting the disease, since it's not certain that they will get it, even if they have the variant. We have another question for you. True or false? If your report says that you have an average likelihood for developing diabetes, it means that you don't have to pay attention to your diet or exercise. This is false. Even though a test may show that you're not at increased risk, you have to remember that lifestyle plays a huge role in complex diseases like type 2 diabetes. If you put too much weight on your test result and lose your healthy habits, you could end up getting the disease anyways. To illustrate this point, let's take a look at another example. This is a report on hereditary thrombophilia by 23andMe. Hereditary thrombophilia is a predisposition to developing harmful blood clots. In this case, Clark did not test positive for any of the results in this test. However, the results show that only the two most common variants were tested. This means that Clark could have a variant causing this disease that is not covered by the test. The test only checks for variants in two genes associated with hereditary thrombophilia and does not give any information on disease risk based on variants in other genes. The report explains that there's still a chance of developing harmful blood clots, and lifestyle and environmental factors can affect your chances of this happening. They also go over some examples of lifestyle factors that can increase your risk, such as a major surgery and oral contraceptive use. If hereditary thrombophilia runs in Clark's family or he suspects he may have this condition, it's recommended that he follow up with his healthcare provider, as this test is not a diagnostic tool.
Okay, so far, the examples we looked at have been carrier status for diseases in one gene and disease risk prediction for diseases influenced by multiple genes. Here we have an example from Medical Genomics, which reports on nutrition and lifestyle aspects. This is a report on the likelihood of being lactose intolerant. Lactose intolerance is the inability to digest lactose or the sugar present in milk. This condition is influenced by one gene. However, unlike the previous carrier status reports, having two risk alleles does not mean you will be lactose intolerant. It is just an estimate because this trait also depends on the environment. If you are more exposed to milk products, your digestive system will become more used to the presence of lactose. When looking at your reports, it's important to understand the type of disease you're looking at in order to understand how much the variants you carry can influence the condition. Here, for the gene MCM6, the risk genotype is CC. In this example, your report shows that you have the TC genotype, meaning you are less likely to get lactose intolerant, but you're carrying the risk allele C for an increased risk for lactose intolerance. If your partner is CC at this position, he will pass down risk allele C for sure. You will either pass down C or T to your kid. If you pass down T, just like you, your children will have reduced risk for lactose intolerance. If you pass down C, your kid will have increased risk for lactose intolerance. The previous examples we looked at show your genotype in one gene or estimated your risk based on two genes. For some health conditions, things are a bit more complicated. One health condition can be influenced by many genomic positions with different variants. One example of this is LDL cholesterol. Here, the company looked at your genotype in multiple genes. All of these variants contribute to your risk of having high LDL cholesterol, which is bad for your health. As you may have already noticed, this company also lists the scientific evidence strands for the variants they tested for. That is to say, how reliable the evidence is for that variant. For LDL cholesterol level, the scientific evidence for how well the markers are associated with the disease is strong, as indicated by 4 stars. They define 4-star strands for large studies with more than 2,000 people who show an association between the variant and the disease.
The third type of report we'll be teaching you about today is ancestry reports. A lot of people are drawn to DTC tests because of this report, so we want to make sure you really understand how they create them and how to understand them. Let's start with some genetic concepts. Every generation, each pair of your ancestors' chromosomes recombined, meaning they crossed over and swapped parts with each other. In the next generation, they separated and paired with another ancestor's chromosomes. The closer two variants are to each other on the chromosome, the more likely they are to move together than separately. So when these chromosomes recombine, these two variants are more likely to end up on the same chromosome than these two. Let's see what happens now, and let's look at just one pair of chromosomes for simplicity's sake. See, I was right. The process then continues every generation until eventually there becomes a small, somewhat fixed stretch of the chromosome that can be expected to always have the same stretch of variant on the same chromosome even after recombination. This is called a haplotype block. A haplotype block is just a linear set of SNPs. By figuring out which haplotype you have at each of these tiny blocks and matching years to the haplotypes from different regions in their database, the company is able to generate an ancestry report for you that's broken down as a percentage of your whole genome. However, keep in mind that it's not a certain record of your ancestry, it's just an estimate. In this example, Jack's DNA matches to regions A, B, and C. Here is an ancestry report from 23andMe. It looks just like Jack's report, right? That's because it's pretty much the same thing, but here it just shows all of the chromosomes. In another report, we see that some values are reported for more specific regions and some are for broader regions. This may be because, according to the databases they're using at the time and the SNPs they choose to look at, the haplotypes in some parts of your genome are only present in one region, like Spain, giving you a specific report, such as Iberian. Other parts of your genome are present in individuals across a broader region, like Southern Europe, giving you a broader report, such as Southern European. There's also usually a small portion that's unassigned. Due to the similarity of the genome sequence across all humans, there are often some regions that are so similar between us that they're unable to say with any certainty where those parts are from. The other possibility is that they don't yet have enough people with your ancestry in their database. More on this later. This is a report from the company Ancestry. They provide maps that show the regions that your ancestors likely inhabited. What's interesting about this map is that the colors don't match the country borders. And that makes sense because hundreds of years ago, the borders we have now didn't exist. We've got another question for you now. True or false? Your results show where your ancestors are from with 100% certainty. This is false. It's super important to understand that the ancestry reports are just estimates. You may see less of a region you were expecting or a whole other region you weren't expecting, and this all depends on the database and the algorithm they're using to generate your estimates. Because of this, your ancestry results will often change every time the company updates their database. We often hear of people getting unexpected ancestry results. This can leave you with a lot of questions and confusion. To clear up some of those issues, I'm going to go through a few reasons why your results could be different from your expectations. As we saw with many of the previous examples, the reference database plays a huge part in the genetic reports, and especially in the ancestry reports. If the database the company used has an underrepresentation of people with your ancestral background, they won't know which haplotypes people from your ancestry have. When they see those haplotypes in your DNA, they won't be able to assign it to any group or the correct group. Here, Matthew knows he's from region G, but he doesn't see it in his report because the database is lacking individuals from region G. He also knows he's from region H, but instead, his report said he was from region E, just because regions H and E are so close together that the algorithm wasn't able to tell them apart very well based on the amount of data the company has at this time. Now we've got one last question for you. True or false? You and your sibling can get different ancestry estimates even from a very accurate test. This is true! Even though you and your siblings may share the same parents, you get different parts of your DNA from each parent. Let me go through an example with you now. Matthew was told that he has some ancestry from region C, but his very accurate ancestry results don't reflect that. Were the stories his grandmother told him as a child wrong? No. In Matthew's case, this is simply a case of recombination acting in a way where the segments from region C are no longer present in his genome. Later, his sister gets a DTC test done, and she does indeed have DNA from region C. Let's see what happened here. Matthew's great-great-grandmother was indeed from region C. After she married his great-great-grandfather, the region C chromosomes recombined with the region G chromosomes in his great-grandmother. This continued with his great-grandparents to his grandmother, his grandparents to his father, and finally with his parents to Matthew and his sister. In the end, Matthew's sister ended up with sections of her genome from region C, and Matthew didn't, and it was all because of recombination. You can also see that Matthew ended up with DNA from 2 other regions that his sister didn't inherit. However, they both share most of their DNA from the same region, region B. Another reason your ancestry report might not line up with what you know is because the test you've chosen may look at where your ancestors were more than several hundred years ago. If your ancestors only moved to the regions you were expecting on your report more recently than that, that may not be reflected in your report. Matthew's back to provide us with one last example. Matthew is from region D, and he knows his family has been there for generations. However, when the test came back, it said he was from region B. Matthew's ancestors likely moved from region B to region D hundreds of years ago. However, the test he chose is only able to look at ancestry from before that. As more data is added to databases, some companies are able to provide ancestry estimates for more recent ancestry as well as ancestry from more than several hundred years ago.
The last type of testing result we will be talking about is traits. A trait is a visual, physiological, or behavioral characteristic of a person. For example, eye color, skin color, and hair type are all affected by our genes. Other less obvious examples are ability to absorb nutrients, caffeine metabolism, weight loss response to exercise, and even whether you like to snack or not. Genetics does play a role in defining who we are, but hold on a second. These traits are more complicated than we thought. Remember what we said about GWAS? Similarly to common disease, many of our traits are affected by multiple genes or variants acting together. Thus, GWAS is a very common method to identify variants responsible for the traits we possess. However, although GWAS may find hundreds of variants associated with your trait, the DTC companies may only include a few of them on the test, so they may not capture enough information to give you the most accurate result. In addition, many traits are heavily influenced by your environment, so your genetic result may not line up with reality. Now let's look at an example report from 23andMe on an interesting trait, whether you like or dislike the taste for cilantro. Some people love cilantro, while for others, it tastes like soap. For this trait, the company tested 2 markers on the genome. Remember what we said about SNPs? There are markers with IDs that start with RS number, followed by 7-digit numbers. The first SNP tested is an A variant, and the other SNP is its B variant on another position in the genome. People with these 2 variants have higher probability of disliking cilantro. Looking at the report, we will also notice that although the company tested this trait for you, due to some error in the process, your variant may not be determined. Sadly, for this person, you won't know your genotype, but I'm sure you know whether you love or hate cilantro. So in this webinar, we went through how to understand the results from DTC genome testing companies. We explained some basic concepts in genetics, different testing methods, and how the variants tested were chosen. Then we talked about how being a carrier for a recessive disorder can affect your children, how not to overestimate your risk prediction for complex diseases, how to understand your ancestry report, and what your trace report can really tell you. Then we went through a few example reports from DTC companies such as 23andMe, Ancestry, and Pathway Genomics, so you can get a better understanding of what to expect. We hope this webinar provided you with the necessary knowledge to better understand your DTC genetic test report. Thanks for watching.